Okay, class, we're talking about the cytoplasmic organelles. The first organelle we'll talk about is the cell nucleus. The cell nucleus is where your DNA is located. It's responsible for signals and dictates kinds and amounts of proteins to be synthesized. Most cells are uninucleate. Red blood cells don't have a nucleus. Some cells do have more than one nucleus. The two ways a cell can get more than one nucleus when we get into it is to do mitosis without cytokinesis. We haven't talked about mitosis. Or several cells fuse together. Your skeletal muscle cells are multinucleated because several cells with nuclei fuse together, which gave a bigger cell with several nuclei. Okay, as you see here, skeletal muscle cells. Here is a general construct of a cell nucleus. The cell nucleus has a nucleolus. We'll talk about real quickly. It's made up of a double membrane, two cell membranes. And then it has little holes called nucleopores in it. This would be the nucleoplasm, which is a fluid, and also in here would be your genetic material. Okay. The nuclear envelope, should be this here, is a double membrane layer. The outer layer is continuous with another structure we'll talk about called the endoplasmic reticulum. So you have an outer nuclear membrane, an inner nuclear membrane, and an intermembrane space. The pore complex allows large particles to come in and out, like something we'll call messenger RNA later in this. So here would be the surface nuclear envelope. Here are the nucleopores. This is an electron microscope picture. Here would be the nucleopores. Here's the outer membrane, inner membrane, and intermembrane space there are also called the perinuclear cisternae. Cisternae means box or chest. The nucleoli, which are plural, or the nucleolus is one. It contains material, genetic material, and bits and pieces of a developing ribosome. So the job of the nucleolus or nucleoli is to develop the ribosomes, which we'll talk about, and I'll bring that up again. What it is a cluster. There's no membrane around it. It's a condensed area of genetic material and bits and pieces of developing ribosomes. Within the nucleus, you have your DNA. We talked about in the chemistry. Okay. The average human nucleus has a diameter of about 5 micrometers, 5 times 10 to the minus 6 of a meter. 5 meters of a meter. If we stretch out the DNA in a somatic cell, that means all the cells that's not the gametes, it would be approximately 2 meters, 6.5. Now think about this. To put this much length into that small space, this much length into that small space. So DNA has to have several folding patterns to make it very organized to put in there. The way I described it in my own way to try to help some of you is the first action is to cut the 2 meters of DNA into 46 pieces. Now, this is just to kind of help you, which would be 46 pieces of genetic material, or also sometimes called chromosomes. Okay? Each piece would be, let's say, around 2 inches or so. A gamete cell, sperm or egg, only has half the genetic material. Then you start this folding pattern. The first folding pattern is a nucleosome. Now imagine you have this thread. Let's say the thread is the DNA. And then you're going to fold it around some marbles. The marbles would be proteins called histones. That would shorten the DNA, but it would get thicker. It would shorten it, but it would get thicker. That first folding pattern is called the nucleosome. That first folding pattern is called a nucleosome. Then you would take the nucleosome that you have and then start lining it up. What you would do is take the nucleosome and kind of wrap it around your finger in a way. and that would form a chromatin. Then the chromatin fiber could be coiled up to make a loop domain. So I'll show you in a moment. Now the DNA gets shorter, but it gets thicker. It gets shorter, but it gets thicker. Histones are responsible for that first level. So here would be the DNA. You coil it up around some histones. Then you start coiling it up in a circular pattern to make it shorter, as you can see. So chromatin is organized into fibers. And ultimately, you're trying to make it shorter. It does get thicker to put it in that small space. It gets shorter and thicker. Now, when you get ready to do mitosis or meiosis, you take this loop domain, and coil it tighter into what we'll call a chromosome chromatid. You only do that in certain phases of mitosis and meiosis. So most of the time, genetic material is in this phase here. Now, I want you to understand this. We talked about the electron microscope and we talked about the light microscope. Under normal circumstances, a cell has the genetic material in the loop domain. That is too thin. Let me say it again. That is too thin to be seen under the light microscope. That is too thin to be seen under the light microscope. So most of the time under the light microscope, the genetic material is unobservable inside the cell. During mitosis or meiosis, we'll talk about, it gets thicker, and that's when you can see it. So if we were in the class, you'd be looking at DNA, chromosomes, chromatids, but you'd only be looking at it during the phase of mitosis. So you can kind of read on it. Here's another example. Take it, coil it up, making nucleosomes. Then take it, so in other words, when you do this, it would look like a pearl necklace. You'd have your DNA, curl it. Assume you take a pearl necklace, beads, take your arm, and what you would do is wrap the pearl necklace around your arm 6 deep. And then you slide that off of your arm, and when you slide that off your arm, that's a chromatin fiber. Then once you take the chromatin fiber, you start coiling it up like this. That's a loop domain, and that's the last folding pattern, and that's still too narrow to be seen under the light microscope. But then when you get into mitosis, during metaphase, we'll talk about, you can see that. That's a chromosome. That's 2 chromatids making 1 chromosome. 2 chromatids making 1 chromosome. We'll talk more about it. But what's the key to this? DNA had to be folded. You're taking 2 meters to try to put it into like 5 micrometers. Okay. You have 2 kinds of genetic material, euchromatin and heterochromatin. Okay. Under differentiation, now this is real quickly. When cells differentiate, you start out with sperm meeting egg. You have 1 cell. That cell could make anything. That's your first cell to make you. That's called a zygote. Then the cell starts multiplying. Initially, when it multiplies to get more cells, because you got to get a bunch of them, all the cells are just alike. That's what we would call a totipotential stem cell, because they could make anything. Then after differentiation, cells specialize into kidney, liver, and so forth. Now you still have all the same genes in all the cells. So your liver cells have the same genes as your kidney cells. But what's the difference? During differentiation, you close up some genes and leave the others open. The ones you close up tightly, you can't read them. The others you keep open. So this would be, for an example, let's say we were in the face-to-face A&P classroom. Let's say we had file cabinets for every course taught on the campus. Economics, history, social study, everything. But in the A&P room, we'd have all the files for the other courses, but the only course files we could open, the others be fused shut, would be those for A&P. So even though they're in the room, they cannot be read. Even though they're in the room, they cannot be read. So genetic material that can be read, liver material in a liver cell that can be read from the DNA, euchromatin. U means true. Heterochromatin is there, but it's coiled up tight. It's there, but it's coiled up tight, and therefore it's there, but cannot be read. That's the basis of differentiation. Every cell has what we call genomic equivalence. A genome is a complete set of genes in an organism. Every cell, every somatic cell has genomic equivalence. All the genes are there, but some cannot be read. Those that can't be read are called heterochromatin. Okay, how do you close them up? By adding chemicals, methyl groups and acetyl groups, that they're there, but you cover them up with these various biochemicals, and they cannot be read. So differentiation is cell specialization. All cells have genomic equivalence. A genome, again, is a complete set of genes in any organism. The key to differentiation is making some genes readable and some not. The readable were euchromatin and the not readable were heterochromatin. A kidney cell would have some genes readable that a liver would not, and vice versa. Euchromatin, readable genes. Heterochromatin, non-readable genes. How do you do it? You put some groups on it to close it off, even though it's present. Okay, so we go back just to be sure that we understand that nucleus. We understand that nucleus. In that nucleus, let's go back to our original picture. We had a double membrane. We had an outer nuclear membrane, an inner nuclear membrane. An intermembrane space are also called a perinuclear cisternae, that space between the two. We had nuclear pores that stuff can enter and exit. We'll need that later. We have in here would be the nucleoplasm, which is fluid, and that grainy stuff that you're seeing is the genetic material, but it's coiled primarily up in the loop domain. Now, the more dotty grains is heterochromatin, because it's kind of snarled up. Then you got the nucleolus. The job of the nucleolus is to have the products responsible for making the ribosomes. Here would be an example of a nuclear pore, and here's outer nuclear membrane, inner nuclear membrane, and perinuclear cisternae. Okay. So now my next video will be on the ribosomes and another organelle after that.